Unit 5, Section 2, Presumptive Testing. When an unknown stain is found at a crime scene, the first type of testing you need to do is something called presumptive testing, which is you don't know for certain what substance it is. Maybe it's ketchup, maybe it's blood, maybe it's animal blood, not human blood. Maybe it's food coloring and Kool-Aid. So you don't know what it is. So presumptive testing is what you'll do when you're assuming it's blood, but you're not sure. So the first question it's going to ask for us is if it is actually blood. Weird. Then it's gonna then the next test we'll want to do, so we check if it's blood. Then the next test we're gonna want to do is check and see if it's human blood. If it is, then we can do some additional testing, which is things like determining blood type, checking alcohol content, and the presence of drugs. And then that's going to be then followed by what we're going to look at later, which is how did the blood actually get there? When we do presumptive testing, there are three types of common tests that are done. The first is the Kastle-Meyer test, the HEMA test, and the luminol test. The Kastle-Meyer test is where you use phenolphthalein, which is like a pH indicator, and hydrogen peroxide. And then this is going to react with the hemoglobin in blood. The HEMA test reacts with something called the heme group in the blood, and this is going to result in blue-green. And finally is luminol, which is kind of the coolest one because it luminesces, it glows. And luminol comes from the Latin word that means light. Alright, so now in order to determine if something is human or animal, a couple ways to do it. The first is going to be through microscopic observation, which I'm going to show you on the next two slides. And that's going to be checking to see just the shape of the blood cell, large, small, presence of a nucleus, things like that. We're going to look at that. And then also there is a precipitin test, which is something that we can take where actually a precipitin if you're familiar with chemistry, if you remember creating something called a precipitate, which is when two liquids are combined and a solid is produced. So that is what we're going to do is you would take blood, inject it to a rabbit. Obviously, we're not going to be doing this one in class. But it forms a precipitate, and if human blood is present, it'll precipitate out. So animal blood cells. Animal blood cells are going to be large blood cells. The nucleus is going to be very large in these, as opposed to human blood, which is going to be very small. The nucleus is basically invisible under the microscope. And what you'll find is a couple other unique things. Tons and tons of red blood cells. The white blood cells are actually going to appear purple under magnification. Okay, so right here. And they're going to be larger and a lot less numerous than the red. And then the platelets you're not really going to be able to identify for our purposes, but you could be able to identify platelets as well under proper magnification. All right. So the next test you're going to want to do is blood typing. Now, when we talk about blood typing, there are four different blood types. We have type A, type B, type AB, and type O. Those are just the general blood types. We'll talk about the Rh factor here in just a minute. Blood type A has antigen A on the surface, and it will agglutinate with blood type B. So again, as a point of reference, we mentioned this earlier, agglutination is the coagulation, the congealment of the blood, and so that's when it's going to kind of clump up. So if I have blood type A, the antigen on that, if it's combined with B, is going to cause it to turn into like a jelly substance. This is why it's so important that you get the right blood type. Back in the old days, when they used to do blood transfers, if it was wrong blood type and they had no way of testing it, it would result in the death of the person, because as soon as blood type B entered the body of individual A, the blood would congeal, it would clog up their blood vessels, and they would die, so it would actually kill them. So that's why upon the discovery of blood typing, we made a lot of advances in terms of saving lives. So then blood type B is going to result with antigen A. Vice versa, just like these two are the same, just roles are flipped. AB has both on the surface, so will actually not agglutinate with either type A or type B. And then blood type O doesn't have them either. All right, so here we go. This is a little graphic illustration to show you kind of what's happening. So you have the blood type here, the antigen that is present, the antibodies that are present, and then who you can give the blood to and get it from. Okay, so blood type A can give blood to A or AB types, but cannot get it from O or A. So just a interesting little note there. I do expect you to actually have a little bit of this known. So who can get what type of blood type is a good thing to know. So the big ones, type O is considered a universal donor. And then blood type AB is our universal recipient. Okay, so notice everybody can get O, and then AB is the only type that can get blood from everybody. So O is a universal donor, AB is a universal recipient. It's AB, AB, AB. Okay, next. And just as a general point of knowledge, type O is the most common blood typing, AB is the least common, with A coming in a pretty close second. So most people are going to be either type O or type A. I got an A plus in blood myself, which brings me to the point of an RH factor. An RH factor is something we mentioned before about the rhesus monkey. It's basically one other factor. You can either have a positive or negative presence of this RH factor, which will be one more way of specifying to make sure you get the right type of blood, because if the RH factor doesn't match up, sometimes there can be additional complications there. So that's what we're talking about. All right, that's all for this one.